My name is Belle O'Bryon. I've been here at the lab for about 5 years. I'm a bioinformatics researcher in the National Security Division. I started at the lab as a postmaster's, so that was a very good opportunity to kind of try out the lab. And then I got hired on as full staff about 2 years after I started. So today I'm going to be kind of talking about much more experimental design, kind of the application that Sam has been talking about, like how to actually set up an experiment that will work when you're using machine learning. So this is a project that was done here at PNNL by lead researcher Becky Hess. It's in my group. So the question that they're trying to solve was issued by DARPA, which is the Defense Advanced Research Projects Agency. So this is an agency in the US government. It started around the time Sputnik was launched in 1957. So they issue these big questions to the research community to answer problems that they think need to get answered, and then they fund them. So this can be at universities, national labs, places like that. And so the question that they wanted answered is, can we isolate a pathogen and keep its phenotype? So this is in, say, like we're familiar with soil microbiomes, very complicated matrix and set of data. And right now, there's not a great way to get pathogen, is there a pathogen in this soil sample, for example, out. We can do DNA sequencing, but DNA sequencing doesn't tell you about the current phenotype. And then also to do DNA sequencing, you're, splitting open the cell and getting all the DNA out. So you're not preserving the phenotype of that potential pathogen. And it's not very accurate because if you're looking at a microbiome sample, it's very complicated. So lead researcher Becky Hess, shown here in her lab, is a microbiologist here at PNNL, but also in my division, the National Security Directorate. And the way she aimed to solve this problem was by using human immune cells, introducing the unknown bacteria and looking at how the human cells respond to that introduced bacteria. That way you're going to be viewing a phenotype. The bacteria is still present, so then you could go do individual DNA sequencing after you see the phenotype, because bacteria are immensely variable and there's a lot of them, a lot of unknowns. But the human immune response is finite and we understand it decently well. So by using that as your marker of is the immune system being activated in this cell line culture, we can take microscopy images and evaluate, is this bacteria that's being challenged, so introduced to the human cell line, is it activating that immune response? So this project was called the OmniScreen project, and they did use machine learning in it. I'm going to be going through their whole experimental design, but the end result that they were going to be testing and evaluating and actually using this model and this whole experimental design on is Arctic permafrost samples. So kind of similar in complexity to a soil sample, but maybe even more unknown and more variable because it's Arctic permafrost samples. And their goal was to start looking at, are there any unknown pathogens that are in the ice melt as the ice melts more? So they thought very clearly, and they thought about all these problems that Sam had mentioned in his talk about when you're using machine learning in science, what do you need to do? Sam had mentioned that there are 4 main things we need to be thinking about. There needs to be a clear question, which here we do. Is the unknown bacteria showing a pathogenic phenotype? There needs to be enough data. So we can't just collect 2 or 3 samples. We need to collect enough that a machine learning model can actually be trained with that amount of data. We need to have an actual score. We need to have a metric that we can evaluate. We need to have something quantitative to let the machine learning model know it's getting close to the answer. And we need to have that close collaboration between our subject matter experts, here in this case with Becky Hess, microbiologists, and the computer science subject matter experts, the people that know which models make sense. Sam just went over a lot of different models. I'm a bioinformatics researcher. I might not know which one makes the most sense for all these problems. You need to have those people all in the same room talking before you're collecting data. Don't just bring in your computer science people at the end. Sam was saying labeled data is very important, and having those people in the room at the same time in the beginning, you can develop an experiment that will label your data that can actually be used very well to use machine learning as a tool for scientific understanding and discovery. So this is the overall pipeline of their experimental design. Parts 1 and 2, I'm not a microbiologist. I'm not going to be diving into as much, but they were very difficult and did take a lot of work on the part of the researchers. So Becky Hess's design team, along with Enoch Young, who was the machine learning expert lead on this team. The first part is if you have a really complicated sample to pre-select for those phenotypes that we want to be looking at. And so they enriched for these pathogenic phenotypes by using activity-based probes and sub-selected from there. Next, they took each of those hypothesized pathogenic bacteria, challenged them into the human cell lines, and then from there did microscopy imaging of over time of those immune responses of the human cells to the introduced bacteria. And then part 4, which is currently in the future work stage, is taking proteomics data and evaluating if there are different proteins that are being expressed or unexpressed between these different samples. So diving into part 1, these activity-based probes, APBs, these are looking for phenotypes that we already know strongly indicate a pathogenic bacteria. For example, toxin production, biofilm production, and up or down regulation of quorum sensing signals. So we know from known bacteria that these things are common pathogenic markers. And so they were able, again, I'm not going to dive into the biology of this, but it was very hard and took a lot of research to label for those different phenotypes and then be able to select those bacteria out of these really complicated matrices of, say, soil samples, and then be able to take those hypothesized pathogenic bacteria, put it in a 96 well plate, and challenge it against the human cell line. And so these human cell lines, we have different spots in our body that can get sick. Common ones are the gut and the lungs. Different pathogens will, infect those areas of our body differently. And so they did look at 2 different cell lines, gut and lung, and they started off creating this data to create the model with knowns. So this goes back to what Sam was saying about there was a question as, is scientific data labeled or unlabeled? Labeled if you make sure it's labeled. So they took 45 known strains of friends and foes. So commensal non-pathogenic bacteria are our friends and pathogenic bacteria are our foes. And so we know what, once we're collecting data, we know this is a friendly response to a bacteria. This is an immune system response because say it's a bad strain of E. coli. We know that that's all labeled. So they introduced their knowns to the different gut and lung human cell lines, and then using microscopy, they imaged these 96 well plates over time. So this is where the vast amount of data collection starts happening that is labeled because we've introduced known bacteria into the human cell lines. Here's some images of that. So on this side, on let's see, your left side, we have healthy bacteria, human cell lines growing. And we can see on the right side that there are the infected human cell lines. And so we as humans can see that there's some patterns, the rounded cells, they're balling up, there's elongated cells. And what would normally happen is a subject matter expert, so a microbiologist who's very good at microscopy, would look at the introduced, the microscopy images of the human cell line that has some unknown bacteria. They would, figure out the patterns that they know as a subject matter expert and be like, oh, this one's infected, this one's not. But for looking at an entire soil, or in this case, permafrost sample, there's going to be way too many because we're checking over way too many unknown bacteria. And so this is why machine learning makes sense here, because we're wanting to have this be a more high throughput method. And so we as people, especially the microbiologists, can see these patterns in healthy and infected cells. The goal is to train the machine learning model to also see those patterns in healthy and unhealthy cells. So the way they did this, again, getting back to those 4 main things that we need to have a good machine learning model in scientific discovery is that we have a clear question: is this pathogenic or not, this unknown bacteria? They were able to collect 14,000 images, which is pretty good for doing machine learning. It's not just a handful with like a couple replicates here or there. They had 45 different strains. They had a lot of time points. They were able to do this, even data collection, in a high throughput way because of the 96 well plates. Here's an example image of their microscopy, which is tagged with different fluorescents. And what they used for their architecture is called a convolutional neural network, also called a CNN. And so I think earlier there was a question about what is good for image processing. This is particularly good at image processing. I'm going to quickly go over why. I'm not going to get too in the weeds because that'll probably take a good long time. But so a CNN, a convolutional neural network, it's good at looking at patterns. What we're trying to use a machine learning model in this scientific experiment is to look for patterns of, this is a healthy human cell, this is an elongated human cell because it's infected, or all those other traits and phenotypes that we're trying to have the machine learning model like pick out for us. And so the way that a CNN does that is, let's say this shape of the filter. So let's say we're looking for a kind of bendy line, and that's our pattern. Then the CNN will it'll make a matrix of that, being like, here's where you have data, dark line, bright line, that matrix at the top. And then I'll take that pattern that we're looking for of that line, and then it'll take your image. So in our case, these are going to be our microscopy images, but it'll go through bit by bit, and I'll look for, is that pattern in my image? Is that pattern in my image? And what it'll do to do that is it'll do basically matrix multiplication, where you have a higher value if the patterns are matching. So again, we have a quantitative way for the machine learning model to know if it's finding what it's looking for. And so using that with the higher values, meaning, yes, we're finding the patterns, the machine learning model continues on and does a lot more complicated layers that I'm not going to get into. But that's, high level. We're looking for patterns. We're training the machine learning model to look for these patterns for us, specifically looking for healthy cells, infected cells. And so this is a part where, again, this was decided not after data was collected, but before, with Enoch Young, the machine learning expert that was part of this project. So this is a kind of complicated figure. I'm not going to dive into all of this, but this is what the architecture actually looks like. Basically, you start with the microscopy images. It goes and does a bunch of neural net things. You get features out. And so here for us, features are, this is a pattern that is useful. This is a pattern of, say, those elongated cells or the rounded nuclei of the human cells. And so we're starting, the machine learning is figuring out what the microbiologist already knows. And then from that, we're able to get actual scores. So as Sam was saying in his talk, we need to let the model know if it's doing a good job. We need a quantitative metric. And so because we have this labeled data of friends and foes that we're introducing into our human cell lines, we're able to measure and have that labeled data, and the model can be like, oh, these patterns are patterns of healthy cells. These patterns of the images are patterns of infected cells. And they're able, because they're doing this over time, if the machine learning model is finding more of the unhealthy patterns over time, that's what these kind of little graphs here, then we can be like, oh, these cells are getting more sick over time. This is likely a pathogenic bacteria. And then from that information, they're able to give it an actual score of how pathogenic that bacteria is. There are human cell lines. So the next part of this project is to look at proteomics data, which is much more my wheelhouse, as I primarily work with mass spec data in my day-to-day. But what they're able to do is collect, again, because we know the human immune response is a lot more finite than every bacteria ever known in the entire world, get the human proteomic samples from, again, the infected or the human cells that have been introduced to pathogenic knowns, and unpathogenic knowns and start looking at what are the differences between them. And so if anyone's familiar with a principal component analysis or a PCA, I'm seeing nods. This is machine learning. This is pretty common within biology. Again, we have labeled data. We have this is pathogenic, this is non-pathogenic. We know that. And then principal component analysis is trying to figure out, okay, how do these clusters, what is the fewest number of, in this case, proteins, which are our data points that can explain the differences. And so we can see that these cluster, very nicely. And then on the other side, those are on the y-axis, just protein names from the UniProt database. But we can see that this bar plot is trying to evaluate which proteins can explain the biggest amount of differences. And therefore, to us as biologists, those proteins are significant. Those are probably, if we go look them up, proteins that are either like fighting off the bacteria or just or not. We'd have to go and look into them. But it's a good way to narrow down what we should be looking at. So they were able to create this model with all this trained data, known pathogens, known commensal bacteria, and then take real Arctic samples from the permafrost, melt them very carefully, and then do their entire experimental design, do their entire process on them. So first, of course, we isolate the microbes using those activity-based probes, looking for phenotypes that we know to be associated with pathogenic, again, toxin production, biofilm production, up or down regulation of corn sensing signals. And so once those are labeled fluorescently, those are sorted into the 96 well plates with the either human or with the human either gut or cell lines, and then imaged over time using microscopy, and then for the pathogenicity. And so again, that scoring pathogenicity is our machine learning model looking for patterns that say, oh, this is a pattern of a healthy human cell, that bacteria, not a threat. This is a human cell that is being attacked. This is a threat, higher pathogenicity score. So these scores also were able to be calculated because, of course, they had positive and negative controls while you're running this. Again, just because you're doing machine learning doesn't mean you shouldn't not do the normal things we do in biology and have controls. So they had a flavor of E. coli that is pathogenic, E. coli, EHEC. So as that is growing in one of the 96 wells, it's able to be kind of a calibration standard of looking at over time, what are the patterns, so features in machine learning language, of these unhealthy cells that are being seen in the E. coli sample. How do all of our unknowns compare to that over time? Again, it's being used as a reference standard. We're being able to calculate, okay, we have all these other 95 other unknowns. How are they comparing over time in the number of patterns or features that are associated with pathogenic bacteria compared to the E. coli? And they're able to calculate pathogenicity from that. So they were able to get a lot of interesting information on what these patterns are. A huge amount of data was able to be ran through from actual permafrost samples. I have it kind of summarized here, the kinds of measurements that they were able to look at. Overall, they were able to narrow down from a vast amount of bacteria, filtering for phenotypes and then imaging those suspected pathogenic bacteria down to 22 potentially pathogenic when compared to the reference standard, and then go do DNA sequencing from there. And so I think the main takeaway from this project is they did a really good job, bullet point by bullet point, as Sam talked about, making sure their data was labeled, making sure they had enough of it, knowing what kind of model they're going to use, or they go do all of this work and do all these imaging so that they know, oh, we're doing imaging data. Okay, we should use a convolutional neural network because to hit on that fourth point of Sam's, we have the microbiologists, the subject matter experts, and the machine learning experts talking to each other and understanding, this is the data I can collect, this is how I can label it. And the machine learning expert being like, oh, that's an image. This is the kind of model that's really commonly used with image analysis.